Hello, everyone. My name is Mariana Aguilera-Puga. I'm a student at Sinvesta Blanquevio, Mexico, and I would like to present my title project, Machine Learning Guided Discovery of Membrane-Active Antimicrobial Peptides. Bacterial resistance has become an alarming emergency due to their low sensitivity to antibiotics. That is why antimicrobial peptides have become a therapeutic alternative due to their mechanism of action. One of the most studied and not very widespread is the formation of pore in the membrane, but it is currently known that other non-lytic mechanisms are just as effective against pathogens. There are reports where relate the physical-chemical properties and residual composition with the recognition of peptides toward the membrane of pathogens. Our hypothesis established that antimicrobial peptides conserve important physical-chemical features regardless of secondary structure. And with the use of machine learning, we will be able to classify peptides in any of three mechanisms of action: membranolytic activity, intracellular target, or no antimicrobial activity. For this project, we compilate a list of 1,048 peptides that we divide into 4 groups. The first is MDPs, membrane disrupting peptides, MPPs, membrane penetration peptides. The last two make up the MAPS group, and finally the PAPS group, protein active peptides, identify peptides that do not have antimicrobial activity. We calculate 180 physical-chemical properties from 3 different computational packages, which will help us to construct 2 orthogonal binary classifiers to predict a specific mechanism of action. The first model is identified as MAPS versus PAPS, and the second model is identified as MDPs versus MPPs. We apply multicollinearity and statistical study for both groups to identify highly correlated properties in both models. Our results filtered out 54 uncorrelated physical-chemical features for the first model and 49 for the second. Comparing the select properties, we observe that some physical-chemical characteristics, such as isoelectric point, penetration depth, is well correlated with some amino acid composition, helping us to identify possible key residues in membrane recognition. Using the physical-chemical properties, 2 orthogonal binary classifiers were built, obtaining the best model with the random forest algorithm with 89 to 96% of cross-validation accuracy. For the result, our models were able to classify peptide sequence in different mechanisms of action. Here, I present the probability of being MDPs in the different secondary structure, but we realized that there could be a prediction bias toward peptides of alpha helix structure because they are the most representative group among the antimicrobial peptides. In conclusion, our best model could predict class membership of 2 MAPs, MPPs, or MDPs with a high accuracy of 80 to 96 using many physical-chemical features. And our models are showing some preference towards alpha helical and random coil structure. Thank you for listening. Poster number 82, and any question or comments, I will be grateful to hear. Thank you.